All right, I'm back. So now we're ready for part 2 of our discussion on the human immune system, and we're going to talk a little bit about the acquired immunity or the acquired defense. And so we talked about your general, specific frontline defense, which is the innate defense, but now we're going to talk about the acquired defense. And so your innate defense does a great job. I mean, think about the constant just bombardment of pathogens. Like if you really stop and think about all the bacteria, viruses, fungi, all the stuff that's out there that, you know, you're constantly getting on your skin and in your mouth and in your eyes. And in general, most of us don't get sick all the time. That innate defense does a really good job. But every once in a while, that innate defense gets overwhelmed by a specific invader. Something gets in and gets a foothold and starts to reproduce so fast that your innate defense can't handle it. And in that case, you need the acquired response. And so the acquired response is much more specific than the innate response. The cells become tuned to specific invaders. And so the acquired defense is very specific, has very specific cells, but you don't want to spend a lot of energy on those if you're not going to use them. So you kind of keep this in your back pocket until you need it. And then when you have a pathogen that is making you very sick, that's overwhelming your first line of defense, now the acquired defense comes in and becomes tuned to that specific invader. And then this is the basis for immunity, because once your acquired defense responds to that specific invader, now it's prepared for any future invasions by that specific invader. And so the next time that bacterium or virus or whatever gets in your body has an experience with that and can respond to it much quicker. So we'll talk about those details now. So the response is triggered by what's known as an antigen. Well, this is also sort of true of the innate defense. You know, antigens are a particular foreign molecule. Usually it's a protein. Usually it's one of those non-self glycoproteins that's on the surface of cells, right? We've talked about those before and how those kind of proteins are used to identify which cells belong to you and which cells are an invader. And so the specific molecules that trigger this response are called antigens. Sometimes it could be a diseased cell of your own that is recognized as something that needs to be expunged from the body too, right? So the response to this antigen, and central to this idea of acquired immunity, are antibodies. So you've heard of antibodies before, so we want to talk about antibodies in a little bit more detail. Antibodies are proteins, and they're a specific type of protein called an immunoglobulin, and we'll just shorthand write IG for those. And so this is a protein that's found on the surface of white blood cells called beta lymphocytes or B lymphocytes. And so you've got those B lymphocytes, those B cells, and they'll have these antibodies sticking on their surface. Or the antibodies are just kicking it around in your blood and your lymph by themselves, right? There's a couple places. So you've got bound ones and free ones. But the antibodies are what your immune system uses to identify these antigens, and then by identifying the antigens, you identify that there's a pathogen that needs to be attacked. And so if you go back to our chart here of white blood cells, again, you can see this is a different type of white blood cell that can form from those stem cells. And this type of cell is called a B cell or a beta cell, and they mature in the bone marrow. So these all the stem cells are made in the bone marrow, but then some of them go to different places of your body to mature into their final stage. But these beta cells mature in the bone marrow. That's where the B comes from. So now each B cell has one particular antibody on its surface. So they don't have lots of antibodies that they're carrying around. Each beta cell has a specific antibody that it's carrying, right? And each antibody can only bind to a specific antigen. And so again, this has to do with what we talked about all semester, proteins and their shape and being able to match shapes. And so these antibodies don't just respond to any old antigen. Each antibody has a specific shape and can only respond to a specific antigen. Here's the trick though. How do they know what the shape of the antigen is? Well, they don't. It'd be impossible to predict the shape of all these antigens because there's so many pathogens and they're constantly evolving. There's no way your body can predict the shape of an antigen, so there's no way your body can predict what shape to make your antibodies. And so your body takes the opposite strategy. We can't predict what shape to make, so let's make every shape. And so there's billions of different beta cells and antibodies in your body, in your immune system. And so consequently, your body most likely has an antigen for a specific pathogen even before you're exposed to it. Okay, so I'm making these now during the COVID crisis. There's a good chance that each of you has an antibody that will match a protein for this COVID virus. But probably, the point is that you probably have this antibody, but you've only got like one or two of them. And it takes time to make copies of that antibody. And so that's why this virus makes you sick. Now, it's possible that, this virus has evolved so quickly that, or has a shape that maybe you don't have an antibody, but you probably have these antibodies, but you don't have many of them until after you've been exposed to it. So how does that work? Well, let's look at the antibodies again. This is a typical way that we draw the shape of an antibody, kind of a Y. And you've got the heavy chains, and you see they're kind of on the inside, and there's two parts to them, and you got the light chains. And think back to our quaternary structure. What we have here is many polypeptides that come together to form a functioning protein, right? That's that quaternary structure. And you've got the heavy chains and the light chains. And then on each of those, you've got a constant region and a variable region. So you've got a part that is the same for all your antibodies, and then you've got these variable regions that's different for each antibody. And this is how you can have all these different shaped antibodies, and chances are you've got one that matches a pathogen before the pathogen gets in your body. And so how do you get all these, again, there's billions of different antigens. How can you produce billions of different antibodies? There's lots of things that cause these antibodies to have different shapes. The DNA that codes for the variable region is in different spots on different chromosomes. And so it's not one continuous gene. It's different pieces of a gene spread throughout your chromosomes that all can potentially mutate independent of one another. These pieces get transcribed and then assembled into an antibody. And so because all these different pieces are out there and then they're just sort of put together to make the variable region, there's lots of different ways they can be put together. And so each way is a different shape, and so that's a different antibody. The DNA that codes for these variable regions also has a higher mutation rate than your other DNA. So it doesn't, you remember how your DNA polymerase sometimes can catch mutations and fix them so they don't become mutations? Well, the DNA polymerase gets lazy in these regions and doesn't work as hard. And so they have more mutations. And so then again, you can create more different pieces of RNA, more different pieces of polypeptide. You can assemble them in different ways. And so there's lots of different combinations that can come together to make a single antibody. And so you can make billions of different antibodies. And so what you're looking at, those variable regions were transcribed from RNA that was transcribed from several different places on the chromosome and then assembled. So there's many different combinations of RNA, and so many different polypeptides that are assembled in many different ways, and it creates these highly variable proteins. So the genes for antibodies are designed to create a huge amount of variability, and so you have lots of different antibodies circulating, each of which is a little bit different, but each of which might match up to some antigen. This is what they call a shotgun approach, right? You can't predict what the antigen is going to look like, so let's just make as many shapes as we possibly can with the hope that one is going to match this foreign invader. But here's the problem. When that foreign invader gets in your body, you probably already have the matching antibody, but you've only got one or two of them. And so this pathogen gets in, and remember that the acquired immunity doesn't kick in until something overwhelms the innate immunity. And so this pathogen got in, and the innate immunity was not able to control it, and it's reproducing, and it's becoming very numerous in your body. But you've only got one or two antibodies, literally one or two in your whole body that match that antigen. And so what are you going to do? You need lots of copies of that matching antibody. And so you've got the matching antibody, which is good, but you don't have enough because it's not enough to control this pathogen that's infecting you. So how are we going to make lots of copies of a specific antibody? And so that's what I'm saying there. We found a match. Now we need to make a gazillion million copies of that matching antibody. How are we going to do that? Well, that starts in the humoral part of the acquired defense. And so that's where the antibodies are circulating in the blood and in the lymph. Okay, so that pathogen has invaded and it's starting to multiply and become a problem. It has unique antigens on its surface that your body can recognize don't belong to you. So your body can recognize that it is a non-self cell. But how's it going to recognize that? How is it going to detect that? The beta cell has antibodies, and every beta cell has different antibodies. Sooner or later, the beta cell with the matching antibody is going to bump in to one of those pathogens that has the matching antigen, right? There's billion different beta cells with antibodies. Sooner or later, you've got lots of this pathogen. Sooner or later, the correct beta cell is going to find and bind to that antigen, all right? When that happens, the beta cell gets to work. When that beta cell finds its match, then it takes that pathogen or takes the antigen into the beta cell. So when that antigen binds to the antibody, that stimulates the beta cell to engulf them both. Then the beta cell presents the antigen on its surface. And so it's like displaying a trophy. It's like, haha, I found the problem. I found the match. And so it takes in that antigen and puts it on its own surface, right? And so the beta cell is now announcing to the rest of your immune system, hey, this is what we're looking for. This is what we're up against. All right, so eventually that's going to happen. The law of averages. It may take a while, but eventually your immune system is going to find that match, and the beta cell is going to say, okay, here's the match. Here, everybody look at this antigen. And so here we're going to refer back to this figure from your book and study that figure and the different steps, and you can see what we're talking about. So here we've got this microbe, and it's shedding these antigens. So, it's dropping these proteins into the blood, and eventually they match up with their matching antibody, and the beta cell now becomes sensitized when it presents that antigen. And now we call it an epitope. And so now it's an activated beta cell. And so now we're going to start that acquired immunity. Okay, so what's the next step? The next step now requires a different leukocyte, a different white blood cell called a helper T cell. So once that beta cell becomes sensitized, becomes an epitope, now the helper T cell recognizes that and binds to it. And so go back to your chart, and this is another type of leukocyte that comes from that stem cell. And so you can see these helper T cells. Now these cells are born in the bone marrow, like all these white blood cells are, but then goes to the thymus in your chest and matures in the thymus. And that's where you get the T. The T stands for thymus. And so this figure is showing how that helper T cell has bound to the epitope. And so you've got the B cell that's presenting this antigen, and that signals the helper T cell to come and bind. And so when the helper T cell binds to this activated B cell, that activates the T cell. So you see how it kind of one thing activates the other, right? The antigen that matches sort of activates the B cell, and then the B cell that binds to the T cell activates the T cell. So what's the T cell going to do? It's a helper T cell. How's it going to help? It's going to help by producing and releasing these types of molecules called cytokines. And there's a million of these things, and they're super complicated. But they're sort of like hormones. It's a chemical signal. It's a way for these immune cells to talk to one another. And so by releasing these cytokines, this starts to signal other white blood cells to get to work. And so there's lots of these, and they're named like IL-4, IL-5, IL-6, IL-10. IL stands for interleukin. These are leukocytes, right? Interleukin, between leukocytes. These are chemicals that allow communication between white blood cells. Okay. So the cytokines start to go and signal other white blood cells, including that beta cell to which the helper T cell is attached, right? So that beta cell that started this whole thing, that's a special beta cell, right? What made it special? It had the matching antibody. Out of all the billions of beta cells with all the billions of antibodies in your immune response in your body, this one matched this particular virus or bacteria or whatever you're attacking. And so that's a special cell, and the helper T cell is going to stimulate that one beta cell. And what's it going to stimulate it to do? It's going to stimulate it to multiply. And so it's going to begin to multiply rapidly. And so that's what we're showing you again in this figure. The helper T cell releases cytokines, and those cytokines tell that special activated B cell, hey, start making copies of yourself. And so again, go back to this figure from your book, and it shows you the helper T cell bound to the epitope, bound to the activated B cell. And then it shows that helper T cell releasing all those interleukins. And it shows then that activated B cell growing and proliferating. So the helper T cell helps that activated B cell make copies of itself. But what do we say was special about this particular B cell? It has the matching antibody. And so by making copies of that particular B cell, you're making copies of that particular antibody. Well, what do we need to do? We need to make copies of that particular antibody. This is how we're going to do it. And so that's what I'm saying here. So when that B cell gets copied, it makes copies of its antibodies. But these aren't any old antibodies. These are the antibodies that are specific to this particular foreign invader. The B cell won't proliferate, won't start making copies of itself until that helper T cell binds to it and releases those cytokines, those interleukins, that tell it to start making copies of itself. And so, what's the helper T? How's the helper T cell help? It gooses that B cell and tells it to get to work. And, it also, those interleukins signal lots of other cells too. Okay, so now we've got the right B cell and it's starting to make copies of itself. That's what we want. It's going to make copies of itself, but they're going to be of two types. Okay? There's going to be plasma cells and memory cells. And so what they're showing you in this figure here again is that special beta cell making copies of itself. Some of those copies are going to be the plasma cell. Some of those copies are going to be memory cells, and it's going to make lots and lots of each of those. Well, what do those cells do? Well, I'm going to go back to this figure here. The plasma cell makes tons and tons of antibodies, but these are the right antibodies. The memory cell just acts as a memory. The memory cell doesn't produce tons of antibodies, but each one has the right antibody or the matching antibody, but it makes tons and tons and tons of these memory cells. So they're just going to chill in your blood, and there's going to be lots of copies of those in your blood. They're not going to do much else, but they have that matching antibody. The plasma cell is going to create tons and tons and tons and tons of the matching antibody. And so I guess that is what I'm saying here. I kind of got myself confused, but that's what I'm saying here. The plasma cell makes tons and tons and tons of antibodies, but again, they're not random antibodies. We're not shotgunning now. We're making all the same antibody, and it's the antibody that came from the original B cell, which was the one that matched this specific invader. So you can see the specificity of the acquired response. So now you've got tons of antibodies, and you're producing tons of these antibodies that are a specific match for this particular invading antigen. So at the start we had just one, right? But now we've got tons of them. And so we don't make copies of an antibody until we need it. And so, like I said, there's a good chance that you've got an antibody that'll attack this COVID virus if you ever get it. But you've only got one or two copies of it, right? And so hopefully you don't get it, but if you do get it, then hopefully your immune system will kick in time to make tons of copies of that particular antibody. Now, when those plasma cells produce all these matching antibodies, they're not put on the surface of the plasma cell, right? You remember those beta cells were going around with these antibodies on their surface. That's not what they're happening. This is just cranking them out and pushing them into the blood and to the lymph, which again are called the humors. And so that's why this is the humoral response. And so we are just pumping tons of matching antibodies out now. Okay, so what? We still have this invader. We've spent a lot of time figuring out what is the matching antibody. We found that out. Now we've made tons of copies of that matching antibody. Great. So what? How does that help us with this infection? Well, those antibodies, of course, are going to bind to the antigens of the invader. Why? Because they're the perfect shape. We've already determined that. So they're going to go bind to those antigens. So wherever you've got a bacterium or a virus or whatever that has that matching antigen, these antibodies are going to go now bind to it. And so then they're going to help control that antigen in a couple of ways. The first way is they're just going to smother it. By just binding to these antigens on that organism, they can make that organism cease to function. So remember, these antigens are proteins that are on the surface of the bacterium or the virus or whatever. And there's lots of them. But if your antibodies come in and bind to all those proteins, remember, you've got to take things, that virus or bacterium wants to take things in and out of its membrane. But if it's coated with these antibodies, it can't do that very well. So that's one way your antibodies can help take care of this pathogen. Another way is that these antibodies can signal those macrophages. And so the macrophages need to know who to attack and who to engulf and who to eat. One way they can know this is by the antibodies. And so if the antibody is bound to it, they recognize, oh, this is something I should eat, and they eat it. So again, you can see how those macrophages were an important part of the innate defense, but here they're an important part of the acquired defense. The two kind of cross over quite a bit. And so that's what we're showing you here in the last steps of this figure, is that the pathogen is being marked by these antibodies. And so now the macrophage knows to come and eat and destroy that pathogen that got in there. So, again, this is a great way to get rid of foreign invaders, to surround them with a cell and then digest them. But, how does your phagocytes, how do they know what to eat? Well, this is one way. The antibodies signal them. Or, again, the antibodies can just engulf and smother the pathogen. And so that's what I'm saying here, right? The antibodies can either just smother the pathogen, which kills it, or they can mark it for death. And so that is what antibodies do for you. And so that's why antibodies are so important. But you can see why you need a gazillion copies of them, right? Because if you're having an infection, by a virus or a bacterium or whatever, then you've got a ridiculous number of these foreign particles. And so you've got to mark them all. And of course, they're constantly reproducing. And so you need a ridiculous number of antibodies to go and try and mark and smother all these pathogens. And you need antibodies that match this particular pathogen. And so that's why you need to make a ridiculous number of copies. And so either way, you need a lot of antibodies, and that's why it's important to fire up that beta cell. And that beta cell starts making copies of itself, and some of those copies are these plasma cells, and those plasma cells, their whole job is to make lots and lots of copies of the same matching antibody. Okay, and so again, this is where I say how the innate and the acquired have lots of crossover, right? And so the cells work together and complement one another and talk to one another through those interleukins and things like that. All right. So remember that when the B cell starts dividing and making copies of itself, it makes a lot of the plasma cells, and they produce all the antibodies that we need for the active fight. But they're also making a lot of these memory cells. And the memory cells are copies of the original beta cell. And so remember, that original white blood cell had this one particular antibody on its surface, and that was all it did, is it cruised around with that antibody looking for a matching antigen. And so now, but when we started, you only had like one of those that matched this particular antigen. Well, now we're making tons and tons and tons of exact copies of that one, and those are the memory cells. And so you got lots of copies of this one floating around now, and each copy has that matching antibody. So now if that same invader invades again, now you don't have to look as hard to find a match, do you? The first time it invaded, you only had one match somewhere in your body. So it took a while before that matching beta cell matched up and then got fired up. Now you've got a gazillion matches in your body. And so the next time that pathogen comes in, boom, we're on it right away. That's how you acquire immunity. is you're exposed to it once, now you make lots of copies of that same beta cell that matches this one. The next time that invader comes in, you're much more likely to recognize it early, attack it early. You know, you'll get the same response, right? That you'll bind to the antigen, the helper T cell will come in, you'll release antibodies, but it'll happen super quick, and you'll jump on that before that pathogen can reproduce very much. And so that's how you get immunity. You'll produce a lot more matching antibodies a lot quicker and a lot higher numbers the next time you're exposed to that antigen. And so that's kind of what they're showing you in this figure here. That you look at, you've got the antibody concentration on the y-axis and the number of days. And day 0 is when you're first exposed to the pathogen. And so again, look on that y-axis, notice it's a logarithmic axis. So each tick is an order of magnitude. It was 1 to 10 to 100 to 1,000. And so what they're showing you is how that first time you're exposed to the pathogen, then eventually you start to produce antibodies to it. And once you control the pathogen, then you can stop producing the antibodies, but you still have the memory cells. And then they show you around day 28 or so, you get a second challenge. You get a second exposure to that pathogen, but look how much quicker your antibody numbers jump up and much higher the antibodies numbers jump up because of all those memory cells. Well, notice also on this how there's a delay, right? You're exposed on day 0, and in this example, it's not like till day 6 or so when you start to actually detect antibodies in the body. Why? Why is there a delay? Well, again, think about it. The first exposure, you've only got one matching beta cell. And so it's going to take time before that matching beta cell finally happens to bump into the matching antigen. And so that whole time, you know, whatever's making you sick, whatever you're being infected with, is multiplying and dividing and doing all that nasty stuff to your cells. And so that's why you get sick, because you've got this foreign thing that's making you sick. And it takes a while for that one beta cell to actually match up. And when it does, then your immune system kicks in, then you make the antibodies, then you start to finally fight against the pathogen and hopefully get rid of it. But now the next time that same pathogen comes in, you've got all those memory cells that have, instead of having one memory cell, one beta cell, you've got a gazillion beta cells. Next time that pathogen comes in, you match it right away, you make the antibodies right away, you jump on it right away. And so that's when you have immunity. And so again, that's why there's a much quicker and a much higher rise in antibodies through the second exposure. It's because instead of having one matching beta cell, you've got 1 million matching beta cells, all of which immediately fire up, all of which immediately grab a helper T cell, start producing antibodies. So, this is something you're familiar with, but now you sort of understand this is what's happening in your body. So again, here's a figure from your book kind of illustrating this and just showing you the same thing in a different way. And they're showing you the helper T cell that's bound to the epitope, the beta cell that's got the antigen. And again, notice that we're talking here about the humoral response. This is in the blood and the lymph node, in the lymph. And then you see that helper T cell releasing all these cytokines, but you see these cytokines, these arrows are going all over the place. They're signaling all these different white blood cells, but one of the white blood cells that gets signaled is that matching B cell. And so that B cell then starts reproducing itself and making these plasma cells. And then the plasma cells make all the antibodies. Okay, so this, of course, is then why vaccinations work. And so when you get vaccinated, what you're doing is you're exposing the body to that antigen. And so, your body's not going to start producing antibodies until it has an antigen to match to. So you're going to inject that antigen, and then at some point the matching beta cell is going to match up to that antigen, and it's going to kick in this whole process. And it's going to produce tons and tons and tons of matching beta cells. Now, it's going to make, plasma cells, and they're going to make antibodies, and but there's not really a foreign invader for your immune system to really attack, because you're not injecting live antigens, you're injecting an antigen which has somehow been killed or neutralized. And so it's not going to reproduce in your body like a typical infection would happen. So how is it killed or neutralized? Again, this is not my specialty, but a lot of times you can just isolate that protein from the pathogen. So you don't inject, the flu virus into your body. That would make everybody sick. That would be unethical. But you could maybe take the proteins off of that flu body and inject those, because that's really what your body's responding to. That's one possible way. I don't really know how you can kill or neutralize these things. You might be able to genetically engineer a virus that doesn't replicate, that makes all the proteins but doesn't then infect your cells and replicate. There's, again, this is not my specialty, but you can get into more details later. But the point is, whatever you're injecting can't reproduce like a typical bacterium or a typical virus. But it does have the antigens of a typical virus or bacterium or whatever. And so, your body somewhere has a match to that antigen. And it just takes a few days, but then that match finds that antigen, the helper T cell comes in, we fire up, we make tons and tons of copies of that matching beta cell. Now you've got those memory cells, and so then if the real pathogen comes in your body, you've got the matching beta cells, and they can bind to it and jump on it and make the proper antibodies right away. That's why vaccination works, is you're allowing your body to produce matching antibodies so that the second time those antigens get in your bloodstream, you're ready for them. And it's all due to those memory cells. So that's why vaccination works. And so that's what they're showing you again on this chart, is that, in this example, they say primary immunization. That's when you get the shot. And so now you're putting that inactivated antigen into your bloodstream. So all you're putting in your blood is that part that matches the antibody, but it can't reproduce itself. And then eventually your body finds the match, you produce antibodies and memory B cells. The challenge immunization, that's, if that virus or whatever gets in your body, you've already got 1 million matches for its antigen. You got 1 million beta cells waiting for it so you can jump on it. And so, your body kills the pathogen for you. That's what your immune system is supposed to do. Now, it is possible to take antibodies from someone who's been infected and inject them into you to jumpstart the immune response, right? So think about what I just said. In vaccination, we're taking the antigen and injecting it and then waiting for your body to produce its own antibodies. In this example, somebody else has produced the antibodies. They've already been exposed to this. They've produced the matching antibodies. Let's take their blood, filter it, pull out the matching antibodies, and stick it in someone else. And so instead of waiting for your body to produce antibodies, you're taking them and putting them right in. And so you kind of skip those first steps. And so you can, not wait for antibodies. I'm going to put them right in. And so that can work, but it's not very efficient, right? You've got to find these people that have been infected. You've got to filter the blood. You've got to pull out the particular antibodies. Then you've got to inject the antibodies. So that takes time and expense. And also it doesn't last because you're only injecting the antibodies. You're not injecting the plasma cells, which will make more antibodies, and you're not injecting the memory cells, which, can also make new antibodies once the original ones are used up. And so it's not a long-term solution, but it can buy time. And this is a strategy that was used in treating Ebola. You know, Ebola is a very bad disease that works very quickly. And so they've had some success. People who've survived Ebola, let's immediately take their antibodies and put them in someone who's sick and control that virus until their own immune system can start making the matching antibodies. And so that's possible, but you know, it has its drawbacks too. Now, newborn babies get these antibodies directly from their mother's milk. And so when a baby's born, the first few times it breastfeeds, that the breast milk contains tons of antibodies that the mother has produced. Think about it. The mother has been exposed to all kinds of stuff her whole life. And so she's got all kinds of antibodies in her bloodstream for things that are out there. The baby hasn't been exposed to anything, and so it's a very vulnerable stage. And so the first few days of breastfeeding, there's tons of antibodies that go into the baby, and so that can, again, if the baby gets exposed to something, it already has some antibodies to help jumpstart the immune system until that baby can start producing its own antibodies. Okay, so that was the humoral response, right, in the blood and the lymph. Last thing we want to talk about is cell-mediated response. And so the cell-mediated response is your last-ditch effort. This is when the pathogen has gotten inside your cells, so you got to start killing off the infected cells. So we already saw this earlier with the natural killer cells, right? And part of the innate response is that it can, you know, the pathogen's gotten inside a cell, so let's just go poke holes in that cell, in our own cell, and kill our cell and take the pathogen with it. This is the same thing, only it's just more specific. And so, again, this starts with the helper T cells. The helper T cell gets stimulated by an antigen, like it did before. But before, that antigen was on a beta cell. It was on an epitope. This time, we don't have an epitope. All right? It's one of your own cells. It's a different cell, not a white blood cell, that's been infected by a virus. Or maybe it's a tumor cell. Maybe it's a macrophage that's ingested a pathogen. There's something, somehow that pathogen got inside the cell, but that host cell produces the antigens from that pathogen on the outside of its own cell. So it's like what, you know, remember beta cells do this, right? When a beta cell finds its match, it then takes in that antigen and then presents the antigen as an epitope. Now it's not a beta cell that's doing it, it's an actual, just a different cell in your body that's been infected, and it's doing the same thing. Again though, that helper T cell recognizes, oh, this is one of my cells, but it's got an antigen on it. We got to kill this thing. And so the helper T cell does what helper T cells do, releases cytokines. And so it releases cytokines which signal other cells to come and let's come and kill this cell. And so again, interleukin-2, TNF, interferon. And so again, this can help to kill tumor cells. And so, you know, my dad had skin cancer, and one of the things they gave him was interferon to kind of help his own cells kill those cancerous cells. And so that's what they're showing you in this part of the figure, the cell-mediated response. And so the T cell, this time, is stimulated by a pathogen that's been, you know, inside another cell. So it's not a beta cell that's stimulating the T cell, it's a different cell. And so now we're releasing different cytokines that are signaling different cells to do different things. Earlier, those cytokines told this B cell, hey, proliferate, make your antibodies. Now we're activating natural killer cells, right, which are designed to go kill your own cells. Also cytotoxic T cells. And so they have a different job, which is to go poke holes in the membranes of your own infected cells to kill them. Again, these same cytokines signal the macrophages. So again, the macrophages, the big bad macrophages are going to come and gobble up pathogens and gobble up things that need to be gobbled up. So all these cells signaling each other. Again, super complicated. We're only taking a very, broad view of all this. But if you want to really understand, medicine and the body's response to pathogens, this is the kind of stuff you need to know. And so here we're showing the natural killer cells, the cytotoxic T cells, the macrophages, all being activated to come do their work. So that's the cell-mediated response when the pathogen gets into one of your cells. Now, sometimes your immune system screws up and it treats your own cells as pathogens. So these T cells are too aggressive. Right? So again, think about what we just talked about. We talked about T cells that are recognizing your own cells and saying, hey, come kill these cells, right? Well, that's a good thing if those cells are infected. But sometimes your T cells make a mistake and get too aggressive, and that's what can cause an autoimmune response. And so things like rheumatoid arthritis, multiple sclerosis, there's lots of diseases that we know about that are being identified as autoimmune diseases. It's your own immune system attacking cells, which is what you don't want. So, these things aren't perfect, right? And so sometimes if you have, helper T cells that are too aggressive, this is what can happen. So, by understanding this, then hopefully we can understand ways to control these aggressive T cells and control this response. Okay, one other disease that you're familiar with that has to do with the immune system is AIDS. HIV, which causes AIDS, it's a virus, and this is a virus which attacks these helper T cells. And so you can see why this is such bad disease, because those helper T cells, they played a big role in all the, in this immune response. And so anything that attacks those T cells is going to have a very negative effect on your immune system. But it is just a virus, right? So why doesn't your immune system attack this virus? Well, it tries to, but one of the characteristics of HIV is that it has a very high mutation rate. And so it's constantly mutating, and so those antigens that are recognized by your immune system are constantly changing shape. And so it's hard to acquire an immunity to this because there's always new versions, and that's what we're seeing with lots of these viruses. That's what viruses do, is that, again, it's a mutation like any other mutation, and it's changing the shape of these proteins, and so you're in a constant battle to try to hopefully find matches to these new proteins with new shape. And so it's difficult to create a vaccine for this particular virus because it has such a high mutation rate. And so I threw a lot at you, and I understand that, but I think this is terribly interesting, and it's usually interesting to a lot of people. It's certainly something that we all have to deal with, and as biologists, you need to have a deeper understanding of these things. Having said that, I realize that we haven't even begun to scratch the surface of all you can learn about immunology. If you're going to vet school, med school, you're going to learn a lot more about this. But hopefully you've got a general basic understanding. So some of these things that like vaccines and things make more sense to you now. So as always, if you got any questions, you let me know. And that's all I have. So I'll see you later.